Okay, everyone, so this is the video for photosynthesis, which is the sister reaction, if you will, to respiration. We always kind of think of the two of them in a cycle together. And this is going to be a process that's carried out by any organism capable of doing photosynthesis. So in other words, you have chloroplasts of some sort within your cell. So this is not something that humans do. Animals don't do this. But we do have bacteria that do this, and of course plants, that kind of thing. So we can take organisms who are capable of making their own food and use the term autotroph or autotrophic to describe them. So if you are autotrophic, why did I just forget how to spell there for a second? There we go. It means that you make your own food. Now, we can talk about eukaryotic cells or organisms that are autotrophic, and then we also have prokaryotes that are autotrophic. So if you're a eukaryote and autotrophic, you are utilizing the organelle, the chloroplast, to perform the function of photosynthesis or the process of photosynthesis. But if you are a prokaryote that's autotrophic, you don't have any organelles, you don't have a chloroplast. So instead, you are going to use your plasma membrane to do the same process. Okay, so that's why you can be prokaryotic and still autotrophic, or eukaryotic and autotrophic. So organisms that are capable of performing photosynthesis are known as photosynthetic, and autotrophs fall into this category. So they are going to use energy to make their own food, and that energy normally comes from the sun initially, and then they're going to use raw materials in the form of carbon dioxide and water to move different molecules around to actually make sugar. So autotrophs make their own food using water and carbon dioxide, and then also energy. Now, photoautotrophs specifically are going to use light energy as their source. So we can also say, oops, there we go, solar. Why are you moving around? There we go. Okay. So examples of photoautotrophs are plants, protists, cyanobacteria. And they're photoautotrophs because they are specifically using light as their main energy source. Now, there are organisms that don't use light. They use some kind of inorganic molecule, sulfur or something like that, as their initial energy source. So that's why we sometimes make the designation of photoautotrophs use solar energy, light energy. Now, if you can't make your own food, you are known as a heterotroph. So heterotrophs are going to consume organic molecules from other sources, or sometimes they decompose organic molecules. You're eating something else. You're breaking something else down. All right, so here we're taking a look at the photosynthesis equation, and it starts off with the raw materials of carbon dioxide and water and energy from the sun. We do the process of photosynthesis, which requires chlorophyll, and we make the products of glucose and oxygen. So that's our full equation for the reaction of photosynthesis. So here we are comparing cellular respiration's reaction to photosynthesis's reaction. And we can do that because it's all the same parts and pieces. They're just going in two different directions. So what do I mean by that? In photosynthesis, we start with carbon dioxide and water, and we make glucose and oxygen. But in respiration, notice the direction of the arrow. We start with glucose and oxygen, and we make water and carbon dioxide. So the products of one become the reactants of the other. And then there's the energy part. Photosynthesis requires an input of energy to make the glucose, but the breaking of glucose in cellular respiration is going to provide an output of energy in the form of heat and ATP. So now let's take a look at the chloroplast, which is the organelle that photosynthesis occurs in. And if you use what we know about the mitochondrion and the reaction that takes place there as a guide, you can see that the inside of this chloroplast has all of these little chambers or locations, and they're actually really important. They're going to play a major role in the reaction we're about to do. So just like the mitochondria, the chloroplast also has a double membrane. We have an outer membrane and an inner membrane, but that inner membrane and outer membrane look very similar to each other. So the liquid inside the chloroplast is known as the stroma, and then we have these stacks. So what exactly are these stacks? The individual disc, so just one single disc, is known as a thylakoid. So this would be a thylakoid, that's a thylakoid, that's a thylakoid. When they're all stacked together like pancakes, that's how I always think of these guys. They remind me of pancakes. They are referred to as a granum. So one stack is a granum, but if we were to talk about this stack and this stack and this stack, then we would be talking about 3 grana. The stacks, the thylakoids, are actually hollow on the inside. So inside, they have a layer or liquid that we refer to as, not a layer of liquid, but they are hollow on the inside, and that space is what we refer to as the lumen. Okay, so let's go through and highlight the important bits. So like I mentioned, we have those 2 outer membranes. The fluid-filled inner membrane, or the fluid that's filling the inner membrane, is called the stroma. The thylakoids, which are sometimes called thylakoid membranes, are those flattened internal compartments that I refer to as discs or pancakes, because they look like a stack of pancakes to me. And when we stack them all together, we call them grana, or granum singular. Some of them are connected. They're held like the stacks are connected one to the next by tubular lamellae. And the space inside of the individual thylakoid sac is called the thylakoid lumen. So where exactly are these chloroplasts found? Well, predominantly they are found in leaves. So leaves are, you can think of them as an organ for a plant that perform a specific function, and they're well adapted to the function that they perform. So I know we tend to think of leaves as just these thin green things that we don't want wilting or turning brown on us, but if you could cut the leaf open and look in on it, it actually has a very complicated structure on the inside. And everything, all the features of the leaves is designed to optimize the gathering of light, and then also the entering and sometimes exiting of the various gases either needed for photosynthesis or made by photosynthesis. So specifically, we have a vein labeled. I'm going to relabel another one right here for you. That structure is a vein, and the vein carries water and sugar. So some parts of it carry water, some parts of it carry sugar. So that's how the water is going to get in and out of the leaf needed for photosynthesis. Then down here we have the stoma, also known as stomata, and that's literally the physical opening or hole at the bottom of the leaf. And this is how your gases get in and out. Okay, so specifically, carbon dioxide is going to enter the leaf this way, and then oxygen, which is made during the photosynthesis process, is going to leave the leaf via that stomata. So where exactly are the chloroplasts? So see this whole area that's labeled palisade mesophyll, this region with these long cells, that's where your chloroplasts are found, the majority of them. So photosynthesis takes place in two major stages or major reactions, and they are known as the light-dependent reactions and the light-independent reactions. So your light-dependent reactions require energy in the form of sunlight. They cannot do their job if they don't have light available. That's why they are light-dependent, okay? So they must have energy from the sun, and they utilize that energy to make, or to not make, but to, sorry y'all, my allergies are killing me and I am just blanking. So because we don't create or destroy energy, we just change its form, that light energy from the sun is going to get changed into chemical energy. And it is going to be used to make ATP and NADPH. Now, NADPH might sound familiar because in cellular respiration, we talked about NADH. So the two of them do a very similar function, but we will expand more on that in a little bit. Now, this light-dependent reaction can sometimes be called non-cyclic and cyclic photophosphorylation. I know that's a big word, but if we break it up, photo for light, and you know the term phosphorylation. We use it in cellular respiration. It's the moving of a phosphate. So movement of a phosphate group controlled by light. That's what photophosphorylation is. Some people also just keep it very simple and refer to them as the light reactions. They occur inside the thylakoid. Okay, you're going to want to make as much chemical energy as you can, so it makes sense to perform these reactions in the part of your chloroplast that has the most surface area. All of those little thylakoids are just like the folds of that inner membrane of cell that happens during that's used in cellular respiration in the mitochondria. We are there to increase surface area. Now, at the end of the light-dependent reactions, the products that you make, what you're forming, is ATP and NADPH. Okay, so when it got imported, my slide formatting changed. I don't know why it does that, but it does. I'm so sorry. Hopefully you should be able to see the proper slide on your end, so I'm just going to kind of go over the information and tell you what to highlight. So the second set of photosynthetic reactions are known as the light-independent reactions or the Calvin cycle. So please highlight that heading. Now we're going to focus in on the words light-independent, which means that these reactions can happen without light. It doesn't require light. If light's there, cool. If light's not there, also cool. Okay? So in these light independent reactions, or in the Calvin cycle, we're going to go ahead and store extra or excess energy by building a sugar molecule, because a sugar molecule is a high energy molecule. It holds a lot of energy because of the bonds that it has. And we can use that sugar molecule to feed ourselves even when there isn't sunlight available, pretty much. Okay? So this is also called the Calvin cycle, and it happens in the liquid part of the chloroplast, the stroma, because it doesn't require a lot of surface area in order for it to function. So in the Calvin cycle, we're going to take the ATP that we made in the light-dependent reactions, and we're going to take the electrons that NADPH, because it's an electron carrier, is carrying, and we're going to use those things to make glucose from the raw material carbon dioxide. Okay, so you should highlight Calvin cycle, highlight stroma, and highlight that ATP and free electrons are used to make glucose from carbon dioxide. So this diagram shows that the two stages of photosynthesis are actually linked together, light-dependent reactions and light-independent reactions, and they do occur at the same time as long as raw materials from the light-dependent reactions have already been made. So as long as I have ATP and NADPH, I can run the Calvin cycle or the light-independent reactions, okay? And the reason they're linked together is because of NADPH and then ATP or ADP because we have the need for those materials. So they just go back and forth. So as you know, light is a very important part of the process of photosynthesis. And light itself is a part of or a form of, the electromagnetic spectrum. It's electromagnetic radiation because it's coming directly from the sun, which is in space. So there's only a small section of the entire spectrum that's actually visible, usable light. And that's the part that we know or we refer to as Roy G. Biv. Okay, so in terms of your spectrum, going from the waves that have the most energy to the waves that have the least energy, gamma rays are going to be the shortest waves, but they have the most energy. And then radio waves are the ones that have the lowest energy. So they're the longest waves, but lowest energy. And the visible light spectrum kind of falls not exactly in the middle, but it falls in between ultraviolet radiation and near infrared radiation. Okay, so in terms of energy, the shorter the wavelength, the more energy it carries. So like the 400 waves over on this side of the spectrum carry the most energy. The longer the wavelength, like these 700s, the lower the energy. So that's the order. Okay, so we do want you to memorize the order of colors, and you also need to know, well, where is high energy versus low energy. So let's delve into this just a little bit more. So visible light ranges in terms of wavelengths. Sorry, adjusting my highlighter. From about 400 nanometers, which is your violet region of the spectrum, up to 700 nanometers, which is the red region of the spectrum. Okay, and that's for visible light. Photons are the units of energy that are carried in light. So that's where the term photosynthesis comes from, because it's based off of the energy in light, which are measured in photons. You really need to know that the shorter the wavelength, the greater the energy. Okay? And over here on this picture, you have a bit of a visual to help with what I mean when I use the term short wavelength versus long wavelength. So if we look at the purple wave, see how the waves are closer together, like the distance here is closer? This will be a shorter wavelength. This is going to be the light that carries a lot of energy. Versus this red wave, see how the wavelengths are further apart from each other? That would be a longer wavelength, so they carry less energy. Okay, so that's what I mean by short waves versus long waves. Now, it's all well and good that these photons are reaching the Earth's surface and all the good things, but how do you harness that energy? How are plants gathering that energy to use it to actually do the process of photosynthesis? Well, it boils down to pigment. So I want you to think of pretty much the color of most of your plants. They're all green. They're green for a reason. So we'll expand on that in a second. So living material, especially living plant material, have pigment molecules inside of their leaves. And those pigment molecules are able to absorb energy from the sun by literally absorbing the wavelengths. So not all pigments work the same. Some absorb certain wavelengths and reflect others. So that's important for you to know. Pigment molecules can absorb wavelengths, but they can also reflect wavelengths. It depends on colors pretty much, okay? So there are electrons in these pigments, and it's the electron that will be absorbing energy from the light, from the photons in the light. So how does this work? Well, electrons can exist in high energy situations or in low energy situations, what we call excited state versus ground state. The electrons start off in ground state, meaning that they're low in energy, so they have space, if you will, to absorb more energy. Once they've absorbed the energy from the sun, they literally jump up to a higher energy level, what we call the excited state. And they are able to then take that energy in their excited state and transfer it to an acceptor molecule. So in photosynthesis, the electron carriers that we're going to utilize, because again, it's very similar to cellular respiration, but the electron carriers that we are going to use carry electrons from the thylakoid to the stroma. Now remember, the thylakoid is where your light-dependent reactions are taking place, and the stroma is where your light-independent reactions are taking place. So this electron carrier is known as NADP+. That's when it's not carrying electrons. And then when it is carrying electrons, it's known as NADPH. Okay, so just like with NADH and FADH2, these electron carriers go back and forth between their oxidized form when they have space to pick up electrons, and their reduced form where they actually are carrying extra electrons. So in the leaf, there are two major groups, if you will, of pigments that are going to help gather energy from the sun. And these pigments are chlorophyll, which you're probably super familiar with, and carotenoids, and those may be a little bit more obscure. So chlorophyll is the major player in terms of the photosynthetic pigment, and it's found in plants, green algae, and cyanobacteria. Pretty much if it's green, it has chlorophyll. Chlorophyll comes in two forms, what we call chlorophyll A and chlorophyll B. The carotenoids just help us to have a bigger base to work from in terms of what energy can be absorbed. So the carotenoids are what we call accessory pigments, and they expand the range of energy that can be absorbed. So chlorophyll absorbs in a very specific part of the visible spectrum. Carotenoids absorb in a slightly different part of the visible spectrum. With all of them working together, it just increases how much energy these plants or these organisms that are photosynthetic can actually gather from the sun. So your carotenoids come in two forms: carotene, which you might have heard the term beta carotene in reference to like carrots, for example. So carotene is an orange pigment. And then hopefully it's not cut off on your slide, but xanthophylls is the next group, and xanthophylls are yellow pigments. I'll rewrite that word just in case. So if you look over here at the diagram, you'd notice that the amount of these different pigments vary depending on the time of year. So in the spring and in the summer, you are going to have mostly chlorophyll A, followed by chlorophyll B, and then finally the carotenoids in terms of how much is available and how much is actually absorbing in that particular leaf. So in this combination, we are absorbing light in these versions of the spectrum and then reflecting light in this green area. So the green area, because chlorophyll is green, the green area does not absorb as well. It's what's being reflected. But in the fall and in the winter, especially in the fall, when those leaves change color, and you always know when autumn's coming because the leaves go from our vibrant green to these really pretty oranges and golds, but it's because the chlorophyll has broken down. Most of the chlorophyll has broken down. And that leaves us with the carotenoids. So now we're absorbing in a little bit of a different area, and we're also reflecting in a little bit of a different area. So this graph is what we would refer to as an absorption spectrum. And what it does is it shows us the percentage of light absorbed by the different pigments in the different color ranges or wavelengths that light energy corresponds to. So the three pigments kind of absorb in slightly different areas, but chlorophyll, being the main pigment, absorbs in the blue and red wavelength areas most strongly, because that's where the graph is tallest, and then a little bit in the violet areas. So I'm going to use my laser pointer to show this. So we are going to focus on chlorophyll A. Chlorophyll A is going to be the solid line, okay? So if we start over here, we get up to a little higher than 40%, then up to about 70-ish%, and that's all in that purple to blue area. And then we fall once we get to this green wavelength of light, which corresponds to just outside of the 500 area, so let's say about 480. We're at the lowest point all the way through until we get to that orangish area, so just outside of 600. And then we go all the way back up to about 55, give or take, percent in this red area. And then we fall again once we pass 700. Okay, so chlorophyll A specifically is absorbing highest in these violet regions and in this red region. No absorptions happening in this green region. Now, if you look at chlorophyll B, which is the dashed line, we are absorbing highest in this teal-ish area. So just about I guess we'd say 490 for that, give or take. And we get all the way up to about 85% absorption. And then again, we fall in the green and yellow regions. And then there's a little bit of absorption of just about 25-ish, 30-ish% in the orange area, and then we fall again. But see what's happening. Chlorophyll B is supplementing chlorophyll A. Chlorophyll A is high here. Chlorophyll B is high over here. Chlorophyll A is high here. Chlorophyll B is kind of high right here. The regions overlap a little bit, but then we also absorb in areas where the other isn't absorbing. Now look at the carotenoids. Theirs is the dotted graph. So they absorb high enough in the violet region, about 50%. And then we continue to go up in that teal region, probably about 65-ish%. And then again in that greenish area, still about 65-ish%, like the light green. And then we fall, and that's the only place that the carotenoids are absorbing. They are not doing any absorption past this, middlemost green area of the spectrum. But again, we're kind of spreading the wealth. We're absorbing in a bunch of different wavelengths so that we can be really efficient at absorbing light energy. Okay, so now that we understand how the pigments work, let's talk about where they're found and how they're arranged. So these pigment molecules are arranged in these large groupings of complexes that we call a photosystem. And photosystems are found on the membrane of the thylakoid. So they're embedded within that membrane, and they're made up of the pigment molecules and then also some proteins. And so they're spread all around the thylakoid membrane. Every little section of the thylakoid membrane has a photosystem. So if you look at this diagram right here, this is an example of what one of the photosystems look like. So this entire thing is a photosystem. So this exists all around this thylakoid membrane. Every single thylakoid membrane has this. Excuse me. And it's done this way to keep the absorption of light energy really efficient. So in these photosystems, the pigments absorb the energy, and then they start passing it along until they get to a part of the photosystem known as the reaction center. In the reaction center is where you're going to find your chlorophyll, and this is also where you're going to find your electrons to change their state, to make them high energy. So the photosystems are broken up into two major sections, if you will. We have what we call the antenna complex, or sometimes it's referred to as the light harvesting complex. And you're going to have chlorophylls and carotenoids spread all through those complexes. And they're arranged in a certain way to optimize the amount of light that's being absorbed from the visible spectrum. Excuse me. The other part of the photosystem is known as the reaction center. And in the reaction center, we have a pair of special chlorophyll complex molecules that also are associated with proteins. And in the reaction center is where the energy is passed from the pigments in the antenna complex to those excited electrons. So the electrons that are about to change state are actually going to be found in the reaction center themselves. So in organisms that are photosynthetic, there are two photosystems. We call them photosystems 1 and photosystems 2. And the differences between the two are slight, but it boils down to the characteristics of the antenna complexes and the reaction centers themselves. Okay, so photosystem 1 and 2 are two types of our photosynthetic systems or units or complexes that are involved in photosynthesis. And the difference is where they absorb. What is their absorption peak? So remember, they are found within the thylakoid. All around that thylakoid membrane, you're going to find these complexes embedded. Photosystem 1 absorbs in that 700 nanometer range. So we sometimes refer to it as P700. And photosystem 2's peak, its biggest point of absorption, is in the 680 nanometer range. So it is sometimes referred to as P680. So if we look at the diagram, here is photosystem 2, here's photosystem 1. All those little green dots represent your pigment molecules that are capable of absorbing light energy. So this would be my antenna complex over here and over here. And then in the middle, see where these two chlorophylls are? That would be your reaction center. Same thing here. So when the light is absorbed, that energy gets passed from molecule to molecule to molecule until it gets to that reaction center. And we go from low-level electrons, see how they're written down at the bottom, to high-level excited electrons. It's moved up to the top. That physical movement represents the fact that we go from low energy to high energy. Okay, guys, so everything on this slide is going to be vitally important. So we're going to start stepping through the two major reactions that make up photosynthesis. So reaction set number 1 are known as the light-dependent reactions. Sometimes we shorten this and just call them the light reactions. These are going to be the ones that are taking place on the thylakoid membrane. And like the name suggests, they cannot occur if light energy isn't available. So that light energy, for the most part, is going to come in the form of sunlight or solar energy. But for your plants that live inside your house, it's going to come in the form of just your lights, your overhead electrical lights. But they need energy in some format. So the whole point of these light-dependent reactions is to gather enough material to make ATP and NADPH. So the process starts when those pigments that we talked about, chlorophyll A and all the other accessory pigments, chlorophyll B, the xanthophylls, the carotenoids, all of those guys, when they start absorbing light as it passes through the leaf. So as they absorb that light in these, what we call the complexes, they are going to start passing the light energy from pigment to pigment to pigment until it gets into photosystem 2. Now, the order that this happens in is very important as well. So that antenna complex is just going to be gathering light and transferring light from molecule to molecule, and it'll do that until it gets to the reaction center of that photosystem number 2. So remember, we have a chlorophyll A molecule that's sitting in that reaction center ready to receive all of this light energy. So light is going to strike the chlorophyll A molecule, and this molecule is the one in photosystem 2. Photosystem 2 is that P680 reaction center. That's the peak at which it absorbs light energy. And it's going to cause the electrons that's in it that are not yet excited to become excited. And as they become excited, they are going to change energy states. They're going to go from being low energy to being high energy. And then they're going to start moving. And they're moving so that they can be accepted by what we call a primary electron acceptor. So they have to move to get to the electron acceptor. Now, during this process, another process is taking place known as photolysis. Some people call it photolysis. And photolysis is the splitting of water molecules by the energy in light. Okay? So as the plant absorbs water, those water molecules in the leaves get split into hydrogens and oxygen as photosynthesis starts to occur. Okay? Now, why do we need this to happen? Well, the water, the hydrogen specifically in the water, is going to donate the electrons that will become excited to P680. Every time water gets split, more electrons get donated. So for as long as we have light, we are going to have the process of the light-dependent reactions take place. So water is going to break apart. It releases electrons and hydrogen and oxygen. Now remember, oxygen is one of our products of photosynthesis. So this is where it's made. This is how plants produce the oxygen that you and I breathe in and out every day and don't even think anything of it. So once those electrons are passed through all of the proteins in photosystem 1, as they're moving, they're losing energy. So by the time, photosystem 2, sorry, is what I meant to say. By the time they get to photosystem 1, they are no longer excited anymore. They have to be excited again. So more light energy has to come in to get the electrons excited. And this time, that's going to happen in photosystem 1. These are arranged in the opposite order. So they were named for the order that they were found in. So we found photosystem 1 first, and then photosystem 2. But you'll notice on the diagram, they're not where you expect them to be. We'll get to that. So once more light energy has been absorbed, those electrons are going to get high energy again, and they're going to, again, move from protein to protein. Remember, the goal is to get to this electron acceptor, okay? And once we get to that electron acceptor, then we've done part of the major responsibility of the light-dependent reactions. Now, as those electrons are passing through these proteins, they're going through all of these redox reactions, and they're losing energy. Okay, so we go from photosystem 2 to photosystem 1, and then finally to our electron acceptor. So once the electrons get to the end of the pathway, they are going to be picked up by a coenzyme that we call NADP. And when it's not carrying any electrons, it's known as NADP+. But when it is carrying energy electrons, it's known as NADPH. So NADPH is an electron carrier that's made in the light-dependent reactions, okay? Whenever you see that H attached to the NADP, it means that the electron is actually attached. So NADP is that final electron acceptor that we've been mentioning, and it has the ability to carry 2 electrons from the thylakoid to the stroma, where another part of the photosynthesis reaction is going to take place. Okay, so there are a couple of ways So sometimes the movement of these electrons are referred to as the Z-scheme shape. And when you see the picture that's coming up, that'll make more sense. So this whole process is known as photophosphorylation, and it is the equivalent of oxidative phosphorylation that takes place in cellular respiration, which is happening in the mitochondria. Now remember, in oxidative phosphorylation, we're making lots of ATP indirectly. So in photophosphorylation, we are also making lots of ATP indirectly. The end result of the light-dependent reactions are ATP and NADPH. Okay, so this is what the light reactions can look like. I'm going to switch to my laser here. So this is what I meant when I said it looks almost like a Z. Like if you look, you'd have to turn it, but here is my Z, my letter Z. And this is what happens when I go all the way through the process and I don't have to repeat any part. So here is where photolysis is going to take place. Okay, and remember, photolysis is the splitting of water. This is what's going to give us those electrons that we need. And then this is where the oxygen is also made. Okay, so once those electrons enter the reaction center, here, I wonder if I can zoom in on this. Nope, doesn't. There we go. Okay, sorry. Doesn't look like I can easily zoom, so we're just not. Okay, I think I figured it out. Okay, so once we have those electrons are going to go to the reaction center. And all along this reaction center, these pigment molecules have been gathering light energy. So the light energy is going to get past the reaction center, and those two electrons are going to get excited. And when they get excited, they literally rise. They change levels because they are now full energy. Now, from here, the electrons get passed through a bunch of different proteins. Each of these little pink circles, if you will, represent a protein complex. And as they move, a bunch of hydrogens are going to start moving through and filling up the space of the thylakoid membrane. And when they do that, ATP is created. So this process is very similar to what's happening in cellular respiration. So the electrons are moving, we're making ATP, and as they move, notice how they're getting lower and lower on the diagram. That represents the fact that they're losing energy. So they get to P700, which is photosystem 1, and they've lost all their energy. They're no longer excited. So more light energy comes in, and those electrons get excited once more. They change state, and then they start moving through these electron acceptors until we get to the final electron acceptor, which is NADPH. So we do the light-dependent reactions because we need NADPH and we need ATP in order to do the light-independent reactions, which are also known as the Calvin cycle. Okay, so let's talk big picture, Where are these light-dependent reactions happening? They're happening in the thylakoid membrane. What is my starting material? The reactants for this reaction is water, light energy. We need to make ATP and NADPH, so we're going to need the oxidized forms of them, which are NADP and ADP and NADP plus. And you're probably thinking to yourself, wait a minute, we're missing a reactant. Where's carbon dioxide? Shouldn't we have carbon dioxide here? Yes, carbon dioxide is a reactant in photosynthesis, but it's not a part of the light-dependent reactions. It doesn't show up until we get to the light-independent reactions. Okay, so what are the products of the light-dependent reactions? What am I making? I'm going to make oxygen, I'm going to make ATP, and I'm going to make NADPH. And you're probably thinking, doesn't photosynthesis make glucose? Where's the glucose? Well, glucose, again, doesn't become a player until we get to the light-independent reactions. So, quick summary. Light energy in the form of photons is going to strike the reaction center of photosystem 2, which is P680. The photolysis of water is going to take place, where water gets broken up into electrons, hydrogen, ions, and oxygen. The oxygen is going to be released from the leaf through the stomata because we don't need it. Those hydrogens are going to travel to photosystem number 1. The hydrogen ions represent the electrons. As they travel, they are going to lose energy. So when they get to photosystem 1, energy is going to come in the form of light once more. Those electrons are going to get excited, and they're going to start moving towards NADP+. When they get to NADP+, the hydrogens get added on to that molecule, making NADPH. As those electrons were moving between photosystem 2 and photosystem 1, we made ATP by photophosphorylation. Okay, so now we're ready to talk about the second set of reactions in photosynthesis, and these are the light-independent reactions, also known as the Calvin cycle. Now, it might be tempting to want to call these the dark reactions. We have the light reactions, so these must be the dark reactions. But dark suggests that this can only happen at night, which is not true. The light-independent reactions can happen 24 hours a day, okay? So by calling them dark, sometimes we misunderstand when they take place. They happen all the time. So they just don't need light. Can they happen in the presence of light? Yes. Can they happen without light present? Yes, okay? So my light-independent reactions, also known as the Calvin cycle, is the process that's going to take the energy that we've captured from the sunlight and store it in a more usable form, which is usually a type of sugar. Okay, so we're going to highlight that. Now, when sunlight is not available, and we're talking about for plants specifically here, when or algae or some of the bacteria that can also do this. When sunlight is not available, that stored carbohydrate gets broken down through the process of respiration so that the plant has energy to do other things, to make ATP, to make NADPH, to make NADH, all the things, right? But we always have to build that supply back up. So the parts of your plant that are capable of doing photosynthesis, aka the leaves, make enough carbohydrates to share with all of the plant, including the parts that are not capable of doing photosynthesis. So this is how the plant ensures that all of its cells has access to energy. Now, in the Calvin cycle is where we utilize the reactant carbon dioxide. And specifically what we're going to do to carbon dioxide is we're going to reduce it. We're going to add in a bunch of other electrons and molecules and change it into this organic material. That's a sugar. Okay? So where are all the parts and pieces coming from? Well, NADPH is going to give us the electrons and the hydrogens to help us make glucose. ATP is going to give us the energy needed to make the bonds to hold all the elements together. And the carbon comes from carbon dioxide. Okay, so this is the way it works in the majority of plants, and we call those plants C3 plants. There are a couple of exceptions to the rules, specifically C4 plants and another set of plants that are called CAM plants, where they don't follow this exactly. They do something slightly different depending on where those plants are found. So that process of carbon dioxide getting pulled into the carbon cycle and starting to become usable for building glucose is known as carbon fixation. It involves gathering carbon dioxide molecules, and it requires the presence of a specific enzyme to do so. That enzyme is called rubisco. Okay? All right. So that's your Calvin cycle in a nutshell. So the Calvin cycle is going to be a similar setup to the Krebs cycle. You're seeing a lot of similarities here because life really takes the viewpoint of if it's not broken, do not fix it. Don't mess with what works. So your Calvin cycle has to go through enough turns so that we can make part of a glucose molecule. Excuse me. So every time that cycle goes all the way through, we call it going all the way through a turn, we actually only capture one carbon because carbon dioxide only has one carbon, okay? So each turn of the Calvin cycle only gives us one carbon from one carbon dioxide molecule. So it means that this cycle has to repeat itself 6 times so that we can make a 6-carbon sugar known as glucose. And most often, photosynthesis makes glucose. So it actually happens in halves. The cycle turns 3 times and makes the first half of the molecule, and then it can turn 3 more times and make the second half of the molecule. So half of a carbon molecule is known as G3P, which stands for glyceraldehyde 3-phosphate. And that's actually the end product of the Calvin cycle. Okay? So the Calvin cycle is broken up into 3 steps or 3 sections. Carbon fixation is where carbon dioxide is added to a starting material called RUBP, and that allows us to produce 2 molecules of 3, okay? And you don't, to be honest, you don't have to know all of the specific names, so I'm pointing out the ones that I do care about you knowing. Like you must know Rubisco, you must know RUBP, okay? So after we go through carbon fixation, if you look on the image, carbon fixation is happening right here. So this is how we end up with 2 3-carbon molecules. RUBP on its own is made up of 5 carbons, okay? So when that carbon comes in from carbon dioxide and I add it to the chain, I now have 6 carbons, so I have 3 and 3, okay? Once that happens, carbon fixation is done. We're moving on to the next phase, which actually is the biggest phase, and this is the phase of reduction. So in reduction, the hydrogens and the energy that we made and captured from the light-dependent reactions, this is where they come into play, okay? So NADPH and ATP are used to convert these molecules right here, these 3-carbon molecules that we make at the end of carbon fixation. We're going to add some things to them. We're going to move them around and add a few parts and pieces, really hydrogens and oxygens and stuff, to this molecule, and it's going to give us half of a glucose molecule, which we refer to as G3P. I need you to know G3P. And also to recognize that G3P and glyceraldehyde 3-phosphate are the same thing. Okay, so these molecules give me half a glucose molecule, and once this happens again 3 more times, I will have another half. Okay, so it just keeps turning. So once that's made, here is my product. Here is my organic compound of glucose. I already made my oxygen, so I've made the 2 products from photosynthesis. But the cycle has to reset itself and get ready to do this all over again. So this is where we're going to utilize some of that ATP that we made in the light-dependent reactions. So once these G3P molecules have left, I am left with the 5 initial carbons that I had already. Okay, so I have to go back and remake RUBP so that carbon fixation can take place again. Okay, and so that's how the Calvin cycle works. All right, so as far as the summary of your light-independent reactions, remember this is not happening in the thylakoids anymore. We're now in the stroma of the chloroplast, so just the liquid part. It involves the reactants ATP, NADPH, and carbon dioxide. It is going to make glucose, and then ADP and NADPH, because once we use the extra phosphate and we use the hydrogens, we just go back to the shell forms of those molecules. So in my Calvin cycle, I am starting with carbon dioxide, which gets put into 3PG. 3PG is converted into G3P, and G3P is converted into glucose. They utilize the enzyme Rubisco to do the carbon fixation step, so it pulls the carbon from carbon dioxide and places it in to the original starting molecule, which is RUBP. So here we're going to talk a little bit more about Rubisco. Now, Rubisco is the enzyme that is in charge of the first step of the light independent reactions. It's the enzyme that is going to catalyze the reaction that takes RUBP and attaches that carbon from carbon dioxide onto it. So it takes care of the carbon fixation step. If Rubisco didn't function in that way, there'd be no way for carbon dioxide to get fixed onto RUBP, and then we wouldn't have the starting material necessary to actually make G3P, which leads to the making of glucose. So Rubisco is sometimes touted as the most important enzyme in the world. It's sometimes also called the most abundant enzyme in the world because the majority of plants that do normal photosynthesis uses Rubisco to do that.
Good morning, Mom.
Good morning, baby. Sorry about that, guys. That was my 4-year-old. Real life happening over here. Okay, so I believe I was talking about how prevalent and important Rubisco was, and the fact that without this enzyme, none of us would have a food source, because this is the enzyme that allows organic material to be made for every living thing. Not every, but the majority of living things. There are a couple of plants that don't follow this normal, quote-unquote, pattern of photosynthesis, what we sometimes refer to as C3 photosynthesis, because we're making a molecule that has 3 carbons in it. There are plants that do a method called C4 photosynthesis, and then there are plants that do what we call CAM photosynthesis. So this is a visual of the Calvin cycle, or the cycle that takes place in the stroma, or the light independent part of photosynthesis. You do not need to memorize all of these parts and pieces in great detail. To be honest, the only things I really need you to focus on, I am going to highlight for you. So we're going to focus on the fact that we use NADH to make NADP plus. And that happens a couple of times, because remember, that's why we have to have the light dependent reactions take place first, because that's when the NADH is made. So that's where it's utilized. We're going to focus on the fact that we utilize ATP. So that happens up here and up here. And again, this is why our light dependent parts of photosynthesis have to happen first, because we need to make the ATP in those steps. Okay, we also use ATP over here. And really, your concern is the fact that we are going to use carbon dioxide. Carbon dioxide's coming in. We're going to attach that carbon dioxide to RUBP. It's going to go through this cycle, changing form a lot, and we are going to make G3P. Okay, so those things that I've highlighted in red, that's your focus. So there are times where the system is very, very efficient and we make a lot of G3P. And when that happens, G3P won't only get turned into glucose, it can actually get made into any of our biomolecules, because G3P is carbon-based with hydrogens and oxygens. And if you think of carbs, lipids, proteins, and nucleic acids, they all start off with carbon, hydrogen, and oxygen as their base molecules. And then we add additional things depending on the nature of the biomolecule that we want to make. So just be aware that if we have extra G3P, it can be used as the starting material for pretty much any of our organic material. Okay, so let's go through a quick recap of the 2 stages of photosynthesis. So keep in mind that they're broken up into the light-dependent reactions and the light-independent reactions. So my light-dependent reactions are going to use energy from the sun to convert and make chemical energy, pretty much. And in doing so, it makes ATP and NADPH so that we can actually make our glucose later on. Okay? This light-dependent reaction can be called non-cyclic or cyclic photophosphorylation. Remember, photophosphorylation is the actual steps involved in making the ATP. Some people also just refer to them as the light reactions. These reactions occur in the thylakoid membrane, and their main products are ATP and NADPH. The next step is known as the light-independent reactions, also known as the Calvin cycle. And in the Calvin cycle, we're going to go ahead and take excess energy and store it in the form of high-energy sugar molecules, or G3P molecules, that will eventually be turned into glucose molecules. And we're doing this because there are times when sunlight isn't available. So even when sunlight isn't available, the Calvin cycle can still function and can make glucose. So that bottom part of my slide cut off, so I just rewrote the three things over here really quick. So another name for those light independent reactions is the Calvin cycle. Note we never call it the dark cycle because it can just truly happen at any time within a 24-hour period. It occurs in the stroma, okay, so in the liquid part of the chloroplast. And ATP and free electrons are used to produce glucose using carbon dioxide, and that also involves the use of the enzyme Rubisco. Okay, so pictorially, here is another recap. Remember, both stages of the reaction of the photosynthesis, if you will, are linked together and run at the same time. Remember, the light-dependent reactions is going to make raw materials that we utilize in the light-independent reactions. So my light-dependent reactions happen in the thylakoid. They utilize carbon dioxide, and we use water as one of our raw materials so we can get our free electrons. As we split that water in the process known as photolysis, oxygen is going to go ahead and leave the chloroplast and leave the leaf via the stomata. Those electrons are going to move until we can make both NADPH and ATP. Once ATP and NADPH are made, they will leave the chloroplast, not the chloroplast, sorry, but the thylakoid, and they will move across to the stroma, the liquid part of the chloroplast, and they will become part of the light-independent reactions, which is also known as the Calvin cycle. So it's made up of 3 steps, the most important step being that first step of carbon fixation, where carbon dioxide, our other raw material, comes in. The enzyme Rubisco is going to attach that carbon dioxide to an intermediate molecule we call RUBP. It is going to go through the cycle, and we're going to make bonds and break bonds, and we're going to make G3P, which is our precursor to carbohydrates and any other organic molecules. So now we're going to talk about the concept of photorespiration. And photorespiration is a situation that can occur because of the enzyme Rubisco. So the active site of Rubisco can actually bind carbon dioxide as a substrate, or it can bind oxygen as a substrate. Normally, it's not a big deal because carbon dioxide is more readily available to the plant, so the plant will bind carbon dioxide once it's in higher quantities than oxygen. However, there are circumstances where oxygen levels rise and carbon dioxide isn't as prevalent, and in that case, Rubisco will bind oxygen over CO2. So that creates this concept or this situation of photorespiration. The issue is photosynthesis, when Rubisco binds carbon dioxide, produces sugar. Photorespiration does not. So it's a dead-end reaction. Okay, it's not giving us G3P, which means we can't make glucose molecules. So why would this even occur? Well, it has a lot to do with where these plants are found. If your plants live in quote-unquote normal environments, not too cold, not too hot, just kind of in the middle, then Plants can keep their stomata, the openings in their leaves, open all the time, and oxygen can leave and carbon dioxide can come out because the plant is not in danger of drying out. The more those leaves are open, the more the stomata in the leaves are open, the more water the plant can lose to its environment. Now, if your plant lives in an area where it's very hot, one of the first things it has to be concerned about is how am I going to hold on to the water I have in my leaves? The easiest way to do that is to close the stomata. So one problem fixed. The problem here now arises that if the stomata are closed, when I make that oxygen in the light dependent reactions, the oxygen can't leave the leaf. It's trapped in the leaf with everything else. The other issue is with the stomata closed, the carbon dioxide from the atmosphere outside of the leaf cannot get into the leaf so that Rubisco can do its job. So now I've created a situation where I have low carbon dioxide numbers and I have high oxygen numbers. And this is a direct result of the temperature that these plants are found in. So if plants live in desert climates or just really hot and arid conditions, this is going to be a problem they face all the time. Under these circumstances, these plants will continuously do photorespiration because they don't have a choice. They're going to have more oxygen available and less carbon dioxide available. So by continuing to do photorespiration, they will also not make any glucose, which means the plant has no food for itself, and eventually that would lead to the plant dying. So this situation with photosynthesis versus photorespiration actually allows us to divide plants into categories based on which pathway they are going to need to use. Normal plants that live, and when I say normal, not like the other plants are abnormal, but plants that live in normal conditions, not too hot, not too dry, any of the things, they can keep their stomata open the majority of the time, are known as C3 plants. So they're called C3 plants because in the Calvin cycle, they go through that C3 cycle, that 3 turns of the Calvin cycle. They also produce a 3-carbon intermediate called 3-PGA, and that leads to the production of G3P. So they literally make a 3-carbon compound, so we call them C3 plants, okay? So some examples of C3 plants are rice, potatoes, soybeans, wheat, again, things that you are fairly familiar with. So what about my plants that do live in these harsher conditions? What do we call them? Well, we call them C4 plants. And that's because they go through a slightly adjusted version of light-dependent reactions. So in the C4 cycle, carbon dioxide is first combined with a 3-carbon molecule called PEP. And that takes PEP and makes it a 4-carbon molecule. Excuse me. And so we get the C4 name from the fact that PEP plus CO2 gives us 4 carbons, okay? The other thing about C4 plants is that they use a different form of enzyme, but we will talk more about that in the next slide. Okay, so C4 photosynthesis. Excuse me, I'm so sorry, y'all. So, like I mentioned before, some plants are going to use the C4 pathway instead of the C3 pathway. The C4 pathway just increases the amount of carbon dioxide available as compared to oxygen so that photorespiration is kept to a minimum. Okay, it's known as Cran's anatomy, and it works even when oxygen concentrations are low. So instead of using Rubisco alone, C4 plants also use the enzyme PEP carboxylase. And PEP carboxylase doesn't have Rubisco's issue. PEP carboxylase is going to bind carbon dioxide every single time. It has no affinity for oxygen whatsoever. Okay, so when oxygen levels are high, because those plants were forced to close their stomata, these plants are going to run the C4 pathway. They're going to use the enzyme PEP carboxylase in order to keep their carbon dioxide in a 4-carbon compound until it gets cooler, so at night, for example, and the plants can go ahead and open up their stomata, thereby releasing all that extra oxygen. Okay, so when those extra oxygen concentrations have been dissipated, when they're lower, the 4-carbon molecule is then going to be oxidized to release carbon dioxide. Okay, so think of it as we're storing the carbon dioxide temporarily when my plant has to have its stomata closed. And then once temperature is comfortable again, so again, like at nighttime, that carbon dioxide gets oxidized out of that molecule, and then we send it through the Calvin cycle like normal to bind to Rubisco. So PEP doesn't replace Rubisco, it works alongside Rubisco. Excuse me. So because of all of the extra steps, the C4 pathway needs extra energy as compared to the C3 pathway. Okay? But in the long term, more energy is saved because we did not go through photorespiration, which is a dead end. C4 plants are significantly more efficient than C3 plants when temperatures are above 30 degrees Celsius. So with C4 plants, it's all about location, location, location. I'm going to store my carbon dioxide in a different location until we can get to use it when conditions are more ideal. Okay, so because C4 photosynthesis has that additional step of going through a C4 cycle so that carbon dioxide can be stored, it means that these plants actually need to have a little bit more space in their leaves to accommodate this C4 reaction. So the C4 intermediate is actually going to be transported or incorporated into a section of the plant called the bundle sheath cells. Okay? And the bundle sheath cells are deeper in the leaf as compared to where carbon dioxide is ultimately released. So that's why we tend to say that C4 photosynthesis is all about location. I'm going to incorporate carbon dioxide into an intermediate molecule, and that happens in the mesophyll cells. And then once conditions are ideal and the leaf can go ahead and open up its stomata once more, I'm going to take that intermediate molecule and pull carbon dioxide off of it and then send carbon dioxide to the bundle sheath cells, which is where the Calvin cycle will actually take place. So think of it as having an additional room in your house where you do a very specific task. Okay, these guys are doing carbon dioxide incorporation in the mesophyll, and then Calvin cycle is actually going to happen in the bundle sheath. So this is in the leaf itself. And just for reference, here would be your bundle sheath cells. So this area, this darker green area, and then here are your mesophyll cells, lighter green. And the reason we're changing location is because in those bundle sheath cells, we are going to have a condition where oxygen is less abundant, carbon dioxide is going to be more concentrated, so it reduces the chances of Rubisco going rogue and doing photorespiration instead of photosynthesis. So that's one alternative to C3 photosynthesis. The last alternative is known as CAM photosynthesis. So some plants that follow C4 photosynthesis also make the additional step of running the C4 part, so taking carbon dioxide and attaching it to the intermediate to make it a 4-carbon molecule, and the Calvin cycle, the actual making of G3P, at different times. So I will run one during the day and the other at night, okay? And they do this to even more avoid photorespiration. So in these plants, we call these plants CAM plants. Crashulin acid metabolism is what CAM stands for. And you see it happening a lot in succulent plants and cacti, desert type plants. So they have the ability of capturing carbon dioxide at night because that's when their stomata are open. So they are going to run the C4 cycle then, but then they run the Calvin cycle during the day when sunlight is available to make the ATP and the NADPH for the Calvin cycle to actually work. So can plants open up their stomata at night? That allows them to capture carbon dioxide and allows them to run the C4 cycle. And then during the day, they close their stomata, which allows them to not dry out, and they use the intermediate malate to store carbon dioxide. So we're going to oxidize the malate, releasing the carbon dioxide inside of our chloroplasts, and then that makes sure that carbon dioxide levels are high, oxygen levels are going to be relatively low, so photorespiration again is minimized. So these guys don't have any new cells. There's no additional cells like with C4 plants. It's just a matter of, is it happening during the day or is it happening at night? Okay, and here's a quick little schematic to compare the three side by side. So we tend to think of C3 photosynthesis as normal, where the two reactions are happening in one in the thylakoid, one in the stroma. Carbon dioxide's coming in. We are running the Calvin cycle in the mesophyll cells, and then we are creating G3P and ultimately glucose, and we're done. Then we have C4 plants, where again, the light, nothing affects really the light dependent reactions because they are already affected by light. So as long as the light dependent reactions can make ATP and NADPH, all of these will run just fine. So we're only focused on the light independent section of photosynthesis for these three plants. So with C4 plants, carbon dioxide comes in. We're going to run the carbon fixation, or the carbon incorporation, I should say, step in the mesophyll, and then we are going to move that compound into the bundle sheath and run Calvin cycle in the bundle sheath. In the mesophyll, we are going to utilize that enzyme PEP carboxylase to store carbon dioxide temporarily into that C4 compound, but in the bundle sheath, we're still going to utilize Rubisco to put carbon into RUBP and do the step of carbon fixation. In our CAM plants, we don't have a bundle sheath cell. Everything is happening in the mesophyll, but we are going to separate what's done, certain parts for the day and certain parts for the night. So carbon dioxide comes in at night. We're going to run C4 cycles at night and then pretty much store them until the daytime. When the daytime comes about, still in the mesophyll cell, we are going to go ahead and oxidize the 4-carbon compound, remove carbon dioxide, and have Rubisco go ahead and put it in the Calvin cycle and make glucose. So C3 runs normally, C4 uses different locations, a mesophyll cell and a bundle sheath cell, and CAM plants use different times. Certain steps are run at night and others are run during the day. All right, and that's it for our photosynthesis video. I will talk to you all in the next one. Bye-bye.